Hi, my name is Enrique Villoj Flores. I am a student of the University of Puerto Rico, Rio Piedras campus, and I am an affiliate of Dra. Cristina Velázquez Marrero's laboratory in the Institute of Neurobiology. Today, I am going to be talking about my research idea, which is based on the measurement of medium spiny neurons, sodium and potassium electrical currents, and what can that tell us about their relationship between alcohol-induced mediated plasticity in direct and indirect pathway medium spiny neurons of the nucleus accumbens core of C57BL6J mice. Based on studies like the ones I am pointing to right here with the cursor, we know that long-term potentiation, which is a type of spike time-independent plasticity induced by alcohol in this experiment, was action potential independent, and that long-term depression, another spike time-independent plasticity induced by alcohol in this same experiment, was action potential dependent. Now, in the same experiments, the medium spiny neurons of the nucleus accumbens core of the type of mice previously mentioned were tested in both naive and after a drinking in the dark protocol, which induced an excessive amount of ethanol drinking. They showed that the direct and indirect pathways had different mechanisms of regulating plasticity after alcohol induction. In both naive and alcohol-induced states, direct and indirect medium spiny neurons of the nucleus accumbens core seemed to, some of them favor a long-term depression state and long-term potentiation state in both the direct and indirect pathways. Now, if it is true that action potentials are dependent of induced plasticity and the results shown, then a way that we can corroborate this and shed light on some other mechanisms not that fully understood in the addiction process, especially in the nucleus accumbens brain region, is to study the density or the effects of it on sodium and potassium, which are essential for action potential occurrence or attenuation. Will be measuring the electrical currents on medium spiny neurons of the nucleus accumbens core of the same type of mice used in the previous experiment mentioned. And we are going to, based on previous literature, to our current knowledge, measure the electrical currents and see if they have a positive or negative association, which better said, action potentials occurrence based on long-term potentiation or long-term depression induced plasticity in alcohol and naive medium spiny neurons of the nucleus accumbens core of the same type of mice. This will help us understand if further studies should be conducted on the subunits of sodium and potassium, different types of subunits of medium spiny neurons, dendritic and somatic parts, because like previous studies, like the ones I have in my presentation here shown, mutations of subunits affect not only the potassium and sodium currents, but action potentials too. In the case of potassium, and prolongation, and in the case of sodium, attenuation. Understanding this relationship can help better narrow down and focus research on an area which is not fully understood and help create new medical treatments for prevention and addiction therapies or treatments. Thank you very much. That is all.